Inside every cell in your body right now, massive, twisting molecules of DNA and RNA are continuously operating to maintain your biology. But when we picture these towering double helices, it is easy to mistake them for a single monolithic entity, like a continuous thread of biological wire. In reality, these giant genetic chains are highly repetitive. They are built by billions of individual chemical subunits, linked end to end in a predictable sequence. Biologists call this foundational building block, the single monomeric units of all nucleic acids, a nucleotide. To understand how the architecture of living things actually works, we have to stop looking at the whole building. We need to zoom all the way in past the helix and investigate the chemical anatomy of this single repeating brick. When we isolate a single nucleotide, we find a highly specific organized structure. Chemically, it is composed of exactly three main components. This 3D wireframe breaks the molecule apart to show its internal layout. Right in the center is a pentose sugar. The prefix penta refers directly to its geometric shape. It's a 5-carbon sugar molecule, forming a rigid ring with 5 distinct points. The exact type of sugar located here dictates the entire molecule's destiny. If the molecule is ribose, it builds RNA. But if it is deoxyribose, which literally means it is missing one oxygen atom, it forms the foundation of DNA. The second critical component is the phosphate group, which attaches directly to the edge of that central sugar. You can think of this phosphate group as the structural adhesive. It is chemically designed to latch onto the sugar of the next nucleotide in line. By alternating sugar, phosphate, the molecule forms a resilient, continuous backbone. This structure protects the nitrogenous bases held within the third component. The schematic isolates the third and final piece, the nitrogenous base, and shows how it plugs directly into our established sugar-phosphate backbone. These nitrogen-containing molecules are the actual data carriers of genetics. They sort into 2 distinct structural categories based entirely on their physical size. First, we have the larger molecules called purines. They feature a bulky double-ring structure and include the specific variants adenine and guanine. Next to them are the pyrimidines. By direct comparison, these are much smaller, built around a single compact ring geometry. They include 3 specific chemical variations: cytosine, thymine, and uracil. There is a strict rule governing their distribution. Thymine is used exclusively when constructing DNA strands. If the cell is building RNA, it swaps the thymine out and uses uracil instead. The size difference between these categories ensures structural integrity. When 2 DNA strands link up, a bulky purine must pair with a compact pyrimidine to maintain an exact even width across the entire helix. This physical arrangement is what allows 2 separate strands to fit together. Because the larger purines always find a match in the smaller pyrimidines, the double helix maintains a constant width, providing a stable environment for genetic storage. Watch in this macro animation as these 3 isolated chemical clusters, the sugar, the phosphate, and the base, slam together. They lock into a single, stable, vibrating nucleotide, ready for action. By simply swapping out the sugar variant or substituting a single uracil for a thymine, the molecule alters its function completely, pivoting between RNA and DNA. Once assembled, this single nucleotide duplicates rapidly. It chains together via the phosphate groups by the billions to build long genetic strands. This architecture is universal. From a single-celled bacterium swimming in a pond to a human being, the fundamental 3-part monomer remains exactly the same. 3 chemically simple components, arranged in varying sequences, successfully generate every biological organism across the planet. The versatility of genetic material is a direct result of this 3-part modular system. It allows a consistent chemical structure to store the instructions for all life on Earth.